right, our last speaker of the main meeting is Joni Wallis. She's going to talk about dynamics of prefrontal computations during decision making.
So I'm going to be talking today about how the prefrontal cortex makes decisions. Okay, so the part of the frontal lobe that I'm interested in is the orbitofrontal cortex. So it's the underneath ventral part of the frontal lobe. And the reason that I'm interested in that is that it's a really nice example where we have a relatively close one-to-one mapping between a specific brain region and a specific high-level cognitive process. So patients who have damage to the orbitofrontal cortex have a very specific problem with decision-making. And all of their other cognitive processes, so things like reasoning, intelligence, planning, traditional tests of executive control, all are intact. And what's more, we don't see problems with decision-making in any other area of the brain. So it's relatively rare to see a very specific problem with decision-making. Orbitofrontal cortex dysfunction is also a component of a lot of neuropsychiatric diseases that have an element of impaired decision-making. So things like addiction, mood disorders, eating disorders, and obsessive-compulsive disorder. You can see that on this slide here. So this just shows the areas of reduced cortical volume in depressed patients. And you can see that the biggest changes are really in this orbitofrontal region and also the adjacent cingulate regions. And this slide here shows where the serotonin transporter binding takes place in the monkey frontal lobe. So the serotonin transporter is a target for antidepressant medications, and you can see that most of the binding is taking place in the orbitofrontal cortex and the ventral medial areas. So part of the motivation for our research is also to kind of try and find ways to improve the treatment of these disorders. One of the treatments that's been used over the last decade or so is deep brain stimulation of these structures. Unfortunately, it's had sort of mixed success, but part of that might be that we don't really understand the optimal ways of stimulating a cortical region. So at least part of our research is trying to improve upon these methods. Okay, so what I'm going to tell you about today, there's going to be sort of two parts to the talk. In the first part, we'll look at how the orbitofrontal cortex uses reward predictions to guide decision-making. And then in the second part, we'll look at how the orbitofrontal cortex learns those reward predictions in the first place. So just to give you a sense of the kinds of experiments that we do and sort of our progress up until relatively recently, this is a typical experiment that we'll do. Everything that we do is primate neurophysiology. So we'll train a couple of monkeys to learn a task where specific or arbitrary pictures predict specific reward outcomes. So here we have a set of pictures that predict different probabilities of receiving a reward. Here we have a set of pictures that predict different amounts of reward. And then down here is a set of pictures where the animal has to work to earn the reward. So he has to press a lever a specific number of times. And this is what an orbitofrontal neuron looks like when we're doing this kind of, when an animal's engaged in this kind of task. So we have time along the x-axis, and then on the y-axis, the firing rate of the neuron. And this is all one neuron's activity. And the different colored lines indicate the value of the picture that the animal's going to choose. So he's being presented binary choices, two pictures are appearing on the screen, and he has to select one of them. And so what you can see is that this neuron increases its firing rate as the value of the picture that the animal's going to choose also increases. And it doesn't much matter how we're manipulating the value, okay? It's a relatively abstract code. Doesn't matter whether we're changing probability or payoff or effort, the response of the neuron is very similar. And if we see about half of the neurons show this relationship, this increase in firing rate as value increases, and about half of the neurons show the opposite relationship, so an increase in firing rate as value decreases. So this is pretty much where we'd got to after about a decade of studying orbitofrontal cortex, and it felt to some extent like we kind of hit a wall, and we were having trouble getting at the actual underlying decision-making process. And the challenge is that it's kind of illustrated in this cartoon here. Okay, so here's a guy faced with a pretty common decision between red and white wine, and this guy happens to prefer white wine. And these are 3 different occasions where he goes through the process of figuring out what he wants on that particular time he's faced with the choice. And you can see that each of the 3 times he goes through a very different process, but we don't have any access to that process. We have the output, which is going to be the choice of white wine, but in each case, the cognitive process is going through a different trajectory. And you might say, well, you've got reaction times. You can look at reaction times. Maybe the top choice would be quicker than the bottom 2, but that still wouldn't be sufficient because we wouldn't be able to tease apart these bottom 2 choices. So we're faced with what we're trying to study as scientists. We can't manipulate it because he prefers white wine. There's nothing we can do about that. That's part and parcel of a subjective decision. And we also can't measure it because it's hidden. So when you can't manipulate something, you can't measure something as a scientist, it's going to be challenging. So the approach that we took is to, this is sort of the standard approach that we've been taking all this time. So where we basically do the same thing over and over again across a whole bunch of different trials and then average the neuron's activity because neurons are very noisy on any given trial. And we create those spike density histograms. So that's what we've been doing for about a decade. And so what we decided to do to try and make some progress is to take a decoding rather than an encoding approach. So just to quickly explain, here what you're looking at is a single trial of neural activity. We're now recording from 40 neurons simultaneously. That pattern of neural activity gets projected into a high-dimensional space, and from that you can decode what behavior the animal's engaged in. So in this particular example, this was, I think, actually from a leech. So the red lines are when the leech is crawling, and the blue lines are when he's swimming. And you can see that those 2 behaviors are projected to different areas of space. And looking at those where those patterns are in that space, you can decode what behavior the animal's engaged in. Now at the bottom here, I just put sensory and motor just to remind myself that this is the approach that's been favored by sensory neurophysiologists, particularly in the primate model, in part because it's really easy to just present the same stimulus over and over again to an animal. But this approach has been favored by motor neurophysiologists. And the reason is it's difficult to get an animal to perform the exact same movement over and over again. There's always going to be variability. And so it struck us that as cognitive neurophysiologists, where we have even less control over how the animal's solving a particular choice, we are potentially better served by this decoding approach than by the encoding approach. And so in the first part of my talk, that's what I'm really going to tell you about. So this is the experiment that we did. We took, in this case, we had 8 pictures. 4 of them were linked to getting a primary reinforcer, so a drop of juice, and 4 of them were linked to a secondary reinforcer. It was basically a token that the animal could exchange for juice. And we equated the value of these using, by varying the exchange rate between the token and the juice, so that each of these was, we titrated it so that each level was identical in value. So we had 8 pictures, 4 different values, and 2 different reinforcer types. And so what we then did is we recorded from small ensembles of 10 to 20 orbitofrontal neurons. One of the challenges with recording from orbitofrontal cortex is it's really deep down in the primate brain. It's about 3 centimeters down. So getting a kind of high yield is technically challenging. So this is the best that we could do at the time. And we then trained a classifier. In fact, it was a linear discriminant analysis, and we would present these pictures one at a time, and we would record the pattern of neural activity, and we would train up a classifier to tell us what value the thing that we just presented was to the animal. And because we didn't have a huge number of neurons, we actually threw everything into this decoder, including the frequency decomposed local field potential. Okay, so that worked fairly well. So the chance level would be 25%, because there's 4 different value levels, and the decoder performed at about 40%. So not great, but it was definitely well above chance. But that's not that interesting, right? Because we've trained a decoder to tell us what picture we presented, but we already knew what picture we presented. So, but the interesting thing is what happens when we then presented a pair of these pictures, okay, and then read out what the decoder was doing. And so our intuition is that when the animal's faced with the pair of pictures and it's having to kind of weigh one thing up versus another, we might be able to see that in the dynamics of the decoder output. And so this is what it looked like. So this is 6 trials chosen at random. The x-axis is time from the choice onset, occurring here about 0. The y-axis is the output of the decoder, essentially how confident it is that that pattern of neural activity reflects a specific value. So it's pretty noisy, but you can also see that there are these periods of stability where the decoder is very confident that the neural activity looks like the value of one of the pictures. And we've color-coded this. So the blue is the picture that the animal's eventually going to choose. The red is the picture that he's not going to choose, and the black is the 2 pictures that were not, the 2 values that were not presented on that particular trial. So we have to convince ourselves that this isn't simply noise in the decoder output. So what we did is we defined these stable states as states where there were 4 consecutive time bins, where the decoder was more than 0.5, had a confidence of greater than 0.5 as to the identity of the value that was being encoded by the network at that time. And on average, on an average decision, the decoder would go through about 5 of these states. And interestingly, the state associated with the thing the animal was going to choose was more frequent and longer duration than the state associated with the thing that the animal wasn't going to choose. So already the first indication that this is something meaningful. It's not just noise. And in fact, the unchosen option was more frequent and longer duration than any of the unavailable options, which typically were not reported by the decoder. So then the next thing we did is we looked at what happened to this decoder on trials where the animal chose correctly and trials where he chose incorrectly. So the incorrect ones didn't happen very often, only about 5% of the time. And that was when the animal was choosing something that we knew was of lower value to him. And you can see that on those trials, the decoder actually goes through more state transitions than on those trials where he performs optimally. The other thing we looked at was the relationship to the animal's choice reaction time. So our intuition here is that the animal will, if the decoder immediately reports the value of the thing the animal's going to choose, then the animal should be able to choose relatively quickly. Whereas if the decoder is instead reporting the unchosen option, then that's going to slow down the overall time it takes the animal to reach its decision. And indeed, that's what we saw. So in here, this is our two animals. And what we're doing now is we're regressing the strength of the decoder output of either the picture the animal's going to choose or the thing that he doesn't choose. We're regressing that against the animal's reaction time. And what you can see is that there is a significantly negative relationship between representing the chosen item. In other words, you represent the chosen item and your reaction time will be smaller or quicker, and a positive relationship for the unchosen option. So this gave us confidence that we're looking at something meaningful here. We've got a state that we can decode with single trial resolution from the orbitofrontal cortex that we can use to predict both whether the animal is going to choose optimally and how long it's going to take him to make that choice. So the next thing we can do, because we've now got this resolution of the unfolding cognitive process, is that we can now essentially timestamp the cognitive process. We can ask what is happening in the neurons and even what is happening in the rest of the brain when the animal switches from considering one option to considering an alternative option. So here's an example where we did that. I'm going to show you some spike density histograms, but now time 0 is a switch in the state of the decoder. So these two plots over here are when the decoder is thinking about the thing on the left, and these two plots over here are when the decoder's reporting the thing on the right. And this is now an orbitofrontal neuron that was not included in the decoder. And the different colored lines are the value, is the neuron's activity grouped either according to the value of the picture on the left or the value of the picture on the right. So you can see that when the decoder is considering the thing on the left, the neuron encodes the value of the thing on the left, but doesn't encode anything about the value of the thing on the right. And when the decoder is thinking about the thing on the right, the neuron encodes the value of the thing on the right and doesn't encode anything about the value of the thing on the left. And that's consistent across the orbitofrontal population. So neurons generally encoded the same thing that was being reported by the decoder and didn't encode anything about the alternate option. So what that suggests is we've got a single value signal being broadcasted from the orbitofrontal cortex. And as the brain switches from considering the two options, considering option A, orbitofrontal cortex broadcasts the value of A, and then when the brain switches to consider option B, it broadcasts the value of option B. Now, the next thing we did is, I told you that we threw everything into this decoder, including local field potentials as well as the spikes. And so we could now look at what their individual contribution to the decoder's performance was. We could remove one signal and retrain up the decoder and look at how that affected our results. So this is an example. This is a decoder that was trained up on just using the local field potentials. Now what we're doing is we're actually trying to decode the identity of the picture that was presented. So we're down, the chance is now 12.5% because there were 8 pictures. And you can see that, again, the decoder is doing fairly well. It's performing well above chance, at least. But we can also look in a little bit more detail and say, okay, what kinds of mistakes does the decoder make? And you can do that looking at a confusion matrix where you essentially, down here is what was actually presented, and along here is what the decoder reported was being presented. It's actually not showing up very well on this slide, but anything along this diagonal is a correct identification, and then anything on the off diagonals is an error. And the errors aren't randomly distributed; they actually lie along this diagonal, which you can't see, but you'll just have to take my word for it. Now we can do so. What that means, just to explain that, is that the decoder is correctly identifying the value, but misidentifying whether it's associated with a primary or a secondary reinforcer. Now, if we do the same thing now for the spikes, so now we're getting rid of the local field potential and we're just looking at the decoder performance from the spikes, you can see that the spikes do better than the local field potential. And when we look at the confusion matrix, what we actually saw was that there was a reduction specifically on these off diagonals. Unfortunately, I have a bar graph just to prove that. And so in both animals, our performance of the decoder for spikes relative to LFP was this ability to correctly identify both the value and the type of reinforcer. So the two things are encoding subtly different amounts of information about the predicted reward outcome. And then the other thing that we can do is look at the different frequency bands of the local field potential. So now we're just training up a decoder solely using one frequency band of the LFP. And what we found is that there were only two bands that were really carrying value information. And they were the high gamma and the theta. So high gamma is not that surprising. It's thought to reflect the underlying spiking activity. And if the spikes are carrying all of this information, then we would expect high gamma to also be carrying the information. The theta was a little bit more surprising, and I'll return to that in the next part of my talk. All right, so just to summarize what I've told you so far, we've seen that decision-making involves the orbitofrontal network switching, alternating between states associated with the value of the two options that are currently being offered. Our current kind of worldview of what this relates to is that this is kind of a biased competition model in which the options kind of have to duke it out for representation in the orbitofrontal cortex, with the more valuable option essentially getting a competitive edge in that, in that competition. And that would explain both why the more valuable option tends to be represented more frequently and for longer duration. And then we also saw that this information is largely carried by the spikes and by theta. Just to give you some insight into what we're currently working on, we're now looking at how does this flip-flop get translated into a decision. And what we're doing is we've now switched to these linear micro wire arrays from Plexon. They're the K probe. We're able to get 4 of them down, and this is giving us about 100 neurons a day. And so we can now look at how the vacillation in the orbitofrontal cortex affects things like the response preparation downstream in structures such as the striatum. Okay, in the next part of my talk, I want to tell you about a different set of experiments that we've been doing, looking at how these reward predictions are learned in the first place. How does the orbitofrontal cortex learn this association between this arbitrary stimulus and the reward outcome that it predicts? So for this experiment, we're using a little bit different task. It's again a binary choice task. There were actually 3 pictures. 2 of those are going to appear on any given trial, and they are associated with getting different probabilities of reward. The difference is that these contingencies are continually changing across the course of a session. So what you can see here, this is an entire session. The y-axis is the probability that the picture will lead to a reward outcome. And there are periods of stability where the contingencies are fixed, and there are periods of instability where they're changing. And the animal has to basically keep track, continually keep adjusting its behavior to adapt to these changes. So you can see in this block, for instance, that the pink is the most valuable, the blue's the least, and the green is somewhere in between. Whereas in this block, blue's most valuable, green's lowest, and pink's the one that's in between. And the solid line is the actual probability that the picture will lead to a reward, and the dotted line is the animal's probability of selecting that picture. So you can see he's doing a really good job of adjusting his behavior to the contingencies associated with the picture. In fact, he's adjusting it in basically in just a handful of trials. Now, one of the things we noticed when we were using this task is that we got some very interesting effects in the theta. So both fixation and when the pictures came on the screen, we saw a phase reset in the theta cycle. So you can see that the color here represents the phase of theta. This is trials along the y-axis, and you can see that the colors line up because the phase of theta is lining up across the trials. And that phase reset and that cross-trial phase lock-in lasts for several oscillations of the theta cycle, so a couple of cycles in fixation, and then 2 or 3 cycles during the actual choice. And we really only see that in the theta. So this shows the cross-trial phase alignment in theta, beta, and gamma at the time of fixation and at the time of the pictures coming on. And you can see that it's really only taking place in the theta band. And what's more, it's only taking place during learning. So this just shows the red line is basically what we would expect by chance from shuffling the trials. And the blue line is the cross-trial theta phase alignment. And the gray is when the contingencies are changing. So you can see in both animals, as soon as the contingencies start changing, we see this theta phase locking. And then as soon as the contingencies restabilize, the theta phase locking disappears. So when you see theta, you start thinking about the hippocampus. So we have got some very preliminary data where we've basically recorded simultaneously from orbitofrontal cortex and the hippocampus. We did this with some custom 3D printed grids that allow us to have different orientations of the recording probes within the chamber. So our vertical probes get us down to orbitofrontal cortex, and then a slanted one gets us all the way down into the hippocampus. And when we did that, we saw phase locking between the two areas, particularly in the theta band at the time of fixation and at the time of the presentation of the pictures. So this was all interesting, but we didn't really have a good handle on what the theta was actually contributing to the performance of this task. And it's pretty difficult to find a way to disrupt theta without disrupting a whole bunch of other things that you don't want to disrupt. So this is the approach that we adopted, and basically what we did is we decoded theta in real time and then used the theta signal to apply microstimulation. So this is decoding theta from the orbitofrontal cortex and using the decoded theta signal to apply microstimulation to the orbitofrontal cortex. So basically every time it went above a threshold and got to the peak of the phase, we would apply a pulse of microstimulation. We then looked at what that did to the learning on this task, right? How well were they able to basically, adapt their behavior to these changing contingencies? So you can see that when we're not stimulating, they adapt really quickly. As I said, it was just a handful of trials before they're adapting so that they can respond optimally 70% of the time. Now when we applied the theta stimulation, we really disrupted the animal's ability to do this task. So instead of a handful of trials, they're taking 50 or 100 trials to readapt to these changing contingencies. And interestingly, if we did the exact same manipulation but at the beta frequency, so we're now decoding beta and applying the microstimulation, we again get no effect. And this is a particularly powerful control because we're stimulating at 3 or 4 times the frequency that we're stimulating the theta frequency. So we're actually applying a lot more electrical stimulation to the orbitofrontal cortex, and yet we're not affecting the animal's behavior. Okay, so what do we think's going on? Well, I'll take a moment to just fit this into the context of what we already know about the orbitofrontal cortex. So one of the what appears to happen or one of the most prominent deficits when you disrupt orbitofrontal function, for instance, by an orbitofrontal lesion or through inactivation, is that you specifically disrupt model-based reinforcement learning. And so that's typically assessed through tasks like this one here, which is a 2-stage choice task, which leads to 2 different outcomes, food or water. And the key manipulation on this task is that you can devalue one of these reinforcers, okay? So you can, for instance, give the animal a whole bunch of water and that's no longer valuable to him. Now, if he's learned the task through trial and error, he has to kind of go down that trial and re-experience the water and realize it's not valuable in order to adjust his behavior. But if he understands the structure of the task, he doesn't need to do that. If he has a model of the task, he knows the water's no longer valuable, so he biases his choices down towards the food. Now, it's that kind of behavior that's specifically impaired when you damage the orbitofrontal cortex. So the orbitofrontal cortex seems to be particularly using task models to generate reward predictions. But it doesn't necessarily mean that the orbitofrontal cortex is responsible for building those task models. And in fact, there's some good reason to suspect it wouldn't be particularly useful for that. The neurons don't encode a lot of spatiotemporal or motor information. So our kind of working model at the moment is that this is accomplished through an interaction with the more sort of cognitive regions of the brain, like the hippocampus or the lateral prefrontal cortex. And the orbitofrontal cortex really is specifically using reward predictions to make decisions and to decide the best course of action. But it's also using models of the task in order to improve those reward predictions. So in our very simple task, there is still task structure. You know there are periods of stability and periods of instability. You know that one picture is going to be high, the other is going to be medium, and the other is going to be low. And so all of that goes into learning and solving this task optimally. And if the 2 areas need to communicate, one way to accomplish that most effective is to have them both firing at the same phase. And when we're applying our microstimulation, all we're doing is we're basically kicking the orbitofrontal cortex out of phase with the hippocampus and disrupting this communication. So that's our idea, and these are the experiments that we've got underway at the moment to explore that idea. We're looking at the effects of stimulation at other phases of theta and also looking at the effects of jittered stimulation. We also want to move to test the effects of hippocampal stimulation. And finally, we're moving towards behavioral tasks that explicitly require the use of task models. So tasks more similar to the one that I just described. Okay, so to conclude what I've talked about, so we've seen that our results are still broadly consistent with the notion that the OFC uses reward predictions that are signaled by cues in the environment. So nothing that I've said really speaks against that, but we've been able to drill down and get a little bit more precision at the underlying mechanisms here. So we've seen that decision-making consists of this alternate sampling of the reward predictions that are associated with each option. And we think that the OFC is in part getting these reward predictions by using these task models provided by these other cognitive brain regions. And finally, we're sort of moving in the direction of developing a brain-machine interface that we can use to manipulate this process. So essentially, the first part of the talk was looking at how to read out of the orbitofrontal cortex, and the second part is looking at how to write in. And so one possibility is that you could design a BMI that would detect the presence of specific stimulus outcome associations and then could apply the microstimulation to disrupt that specific association. And that could be useful for things like addiction, where you have a small set of stimulus-reward associations which have seized control of a patient's behavior. So I just want to finish by acknowledging the people that did all of this work. So Erin Rich did the decoding experiments while she was a postdoc in my lab. She now has her own lab at Mount Sinai School of Medicine. And Eric Knudsen has been doing the OFC stimulation studies. I also want to acknowledge our funders. Thanks.
So Antonio Rangel has been modeling the kind of choices you were looking at the beginning of your talk. And he has proposed that you can capture the behavioral data very precisely with a drift diffusion model in which your drift rate depends on which object you're considering, right? Yeah. And there was some parallel with the OSC data that you showed us. There was also quite a few differences, in my opinion, but I just want to get your take on it. Do you think of the OSC results as very much like a neural implementation of the Rangel model?
So I think that, so the Rangel model states that eye movements play an important part in the control of that drift diffusion. We looked at that, so we synced all of our decoder outputs to eye movements, and there was no effect of that. But I do think that you could imagine that a downstream structure listening to these alternations could be integrating those flip-flops. And the fact that the chosen option, the more valuable option, occurs more frequently and it lasts for a longer time, that would be sufficient for the drift diffusion model downstream to come to a decision as to which should be the appropriate response. And in fact, that's what we're looking at now with the simultaneous OFC caudate recordings to see whether that holds up.
Hey, this is stunning data, particularly, well, I know the first set, so the hippocampal stuff looks amazing. So Eichenbaum labs say that the LFP leads, the hippocampal LFP leads the OFC LFP in choice, and then the OFC LFP leads the hippocampal LFP at the outcome time for learning. So firstly, I was wondering if you see it, wondering which one's leading in your phase comparison, and then I was wondering whether it's affecting learning or choice.
I have no idea. So we've only just collected that data. We collected that data like a couple of weeks ago. I stuck it in, and that was just one session.
Okay.
So yeah, really it's been a challenge to get that, do those simultaneous recordings in monkeys. I think those are the first ones.
But you can tell from the microstim whether it's affecting learning or choice, right? You can tell whether it's affecting future trials or whether it's affecting the current trial you're stimulating on.
We can, well, it's just disrupting choice entirely because it's, because the animal's trying to learn. So yeah, it's not done in the steady state.
Thank you very much for the really nice talk. Maybe I missed this, but in terms of equating the value across the primary and secondary reinforcers, are you seeing that the decoders that you're building or the oscillations might be able to distinguish between those as well? Like if you present the two that have the same value, but one is primary, one is secondary, would you be able to see that using your spike decoder, which didn't have those kinds of off-diagonal peaks in the confusion matrix?
Yeah, so we looked at that. So we basically did, we had one idea, which was to build a decoder that looked at reinforcer type, and a second decoder that looked at value and see whether the two flip-flopped at the same time. Unfortunately, there's just not enough information contained about type, reinforcer type, even in the spikes. Okay. It's too noisy to extract that information.
Okay, thanks.
Hello, great talk. I have a question about the relationship between the decoding, the alternation in decoding the different options and attention in the task, and how does it relate possibly to eye movements? Like, is the monkey basically encoding the value to which he pays attention, or is it more an internal process within the UFC which alternates naturally?
Yeah, I don't know, and I've thought about both. So we know it's not eye movement, so it's not as simple as that. And we've also looked at things like microsaccades, they're not predictive. But we can't rule out the possibility that it is like a covert attentional process. So we've had some success in the past at decoding attention, decoding covert attention from lateral prefrontal cortex. So yeah, one of the experiments that we've got planned in the near future is basically simultaneous lateral prefrontal with a spatial decoder and the orbitofrontal decoding.
Okay, I have a second question. There are some data in humans suggesting that there is a spatial dissociation in the OFC between primary and secondary rewards, and it seems that your design could address this question. So did you see any?
We didn't, no. So in fact, the original design was not for this experiment at all. It was in fact for that, to look at that, and we couldn't see anything, no.
Hi, really interesting work. I was wondering if you can comment on what accounts for the variability in behavioral performance after microstimulation in Theta.
Well, so in terms of the effects of microstimulation?
Yes.
No, I don't have a good handle on that. We are doing a lot more now to try and look at neurophysiological. So, for instance, we only had one theta decoding electrode and one stimulating electrode, and so we're going to look at sort of a bigger array of electrodes down in orbitofrontal cortex to see whether, on those days when we're having a bigger effect, we're essentially entraining more of the network to our artificial frequency compared to days when we have a smaller effect. So we have some ideas, but no data.
Okay, because I was wondering if you had mentioned that the model that the animal might be using to solve the task involves hippocampus, right? So if that's not intact, then maybe the animals have to sample the choice more extensively, and if that could be associated with that variance.
Yeah, I mean that's what the task that we've used is not a very good task for looking at model-based reinforcement learning, and so that's on our to-do list, is to use the task where you actually need the model. Not that it's just kind of giving you a little bit boost in learning, but that you actually need the model in order to solve the task, and hopefully then we'll be able to get, pin down more precisely what the deficit is.
Great, thanks.
Great talk. So my question is a follow-up on the last things that you were saying. It would be a great thing if there's a cognitive map in the hippocampus and it's explored to find options and check the desirability with OFC and see how to perform tasks. But I'm wondering, what do you think is driving this internal navigation? So in the model-based condition, it could be that you're just doing some kind of sweeps to see which one leads to some desirable outcome. In this case, it seems like it might be exploration or noise. What do you think is going on in this case? And what is the region that would be driving that? And is it happening in hippocampus, coming to OFC? Or is the OFC not sure and it's a measure of certainty? And so it's vacillating between options in one case, whereas when there is certainty, then...
Yeah, I mean, I don't really know. My suspicion is that probably the most important thing that's being provided is state. Right? In other words, am I in a stable state where I don't really need to be updating my contingencies, or am I in a unstable state, in which case I now need to start changing everything and tracking contingencies? That's sort of my best guess, but I don't have any data for that. And I think we just need a better task for studying the model-based.
And where do you think that certainty comes from? Is that just prediction errors or something else? What is it, striatum that is helping, or some ACC or other regions?
Yeah, I don't have a feel for that. There has been work by Tim Behrens looking at ACC for things like volatility. I know that Sam Gershman has some work on prediction errors, where small prediction errors meant you just had to do a small update, but large prediction errors meant, your model's probably wrong and you need to come up with a new model. So yeah, any of those would be potential candidates.
All right, let's thank the speaker and all the speakers in the session.